Good morning, everyone, and welcome to the Science on the Screen. It is my pleasure today to present Sofia Duarte, who I noticed that I know for more than 20 years now, despite her being so young. She has a degree in applied biology and completed the PhD degree in sciences with us here at the University of Minho. And during her studies, she was mainly focused on the effects of anthropogenic stress, namely eutrophication and metals, on the diversity and activity of microbial decomposers of plant litter in fresh waters. And after her PhD, then she developed postdoctoral studies about DNA barcoding and metabarcoding of aquatic fungi here at the University of Minho as well, but also at Mount Allison University in Canada. More recently, she shifted her interests from freshwaters to estuarine marine ecosystems, and where she was assessing the performance of metabarcoding approaches in diversity monitoring of benthic estuarine communities, in particular of myofauna, in collaboration with the University of Guelph in Canada. In 2019, Sofia became an assistant researcher at the University of Minho, and now presently she is coordinating the NIS-DNA project funded by FCT, which aims to optimize molecular tools for the early detection of non-indigenous species in Portuguese coastal ecosystems. So it is our pleasure to have Sofia Duarte with us today to talk to us about her more recent developments in her research.
So thank you, Professor Celia. I will share now my monitor. So thank you, Professor Celia, again for this invitation, also for presenting me. It is a great pleasure to talk a bit about my work at the Science on Screen. And I have very good memories of Professor Celia because she was the first professor that I met when I started my graduation in applied biology. And for those that do not know me very well, indeed I'm for more than 20 years associated with the biology department from University of Minho. I can tell you, I don't know if in English we say like that, but that I make part of the furniture. And I have been working for long in aquatic ecology. I started with fresh waters. Now I'm working in coastal and marine ecosystems. And since assessing biodiversity, it is so important, in particular during these times of global change, it is important to assess more often diversity and trying to develop tools to assess it faster. And that's why I am being dedicated in the past few years to the development of DNA-based tools in order to improve biodiversity assessments. And today, with this presentation, I will try to answer, if we are ready, to use DNA-based tools to detect and monitor non-indigenous species in coastal ecosystems. So coastal ecosystems are among the most valuable ecosystems on Earth, and they are widely recognized by their high ecological and socio-economic importance. They are also the home for many species, and they are also among the highest productive ecosystems on Earth. And they also provide very important ecological functions and services, such as nutrient recycling, climate regulation, erosion control. They also provide seafood and other raw materials, for instance, that are used in the production of medicines. They are also used for the transportation of goods and services and also passengers, and they are also widely procured for leisure activities. But these ecosystems are also exposed to major threats such as climate change, overexploitation of resources, pollution, habitat
Sofia,
destruction.
sorry to interrupt. I think that we are not seeing your presentation.
Oh, sorry.
Can you try to share the screen again?
Yeah,
No, it's
of
okay.
course. And now?
Now we are.
Okay. So I will put here.
It's okay. We are in time. Don't worry. Yeah. Okay.
You are seeing?
Now it disappeared again. Can you try?
It appeared, and now?
No. Can you?
I don't know what is happening. Let me see. And now?
No.
Oops. No. I don't know because maybe let me see.
Sofia?
I have the VPN connected. Maybe
because we tried before the meeting start, we tried and it was working okay, so don't worry.
I will try again.
Okay, we are seeing it now.
Okay, let me just put in the
Presentation.
Okay, presentation and you. Okay, right. Now it's okay?
Yes.
Okay, as I was telling to you, these ecosystems are also exposed to major threats such as climate change, overexploitation of resources, pollution, habitat destruction, and also the spread of non-indigenous species, which can compromise the integrity of marine and coastal ecosystems and the goods and services that these ecosystems provide. And in particular, non-indigenous species, it's a major threat to coastal ecosystems, and these species are species that are introduced in areas outside their natural occurrence range. And the problem of these species is that they can spread rapidly and become invasive, displacing and outcompeting native species. By competing by the same food, also by the same habitat, or even predating on native species. And this can conduct to diversity loss and alterations in ecosystems. As you can see here, this site completely invaded by an invasive algae. And these species, due to their massive growth, they can also provoke severe economic impacts in countries. And once established, they are extremely difficult to eradicate. And among the 100 worst invasive species, 32% are aquatic marine, which is a significant proportion. But how do these species arrive into coastal ecosystems? They arrive namely through shipping. They get right on the ships, namely through ballast waters discharges that are waters that are used for the ships for maintaining the equilibrium in the sea to compensate the cargo, and by collecting these waters in a different geographic location from the one of destination, they can transport in this water that is released then in the destination ports. They can transport organisms alive. Also, a major vector due to shipping is through biofouling, where the organisms incrust into the ship structures. And ballast waters discharges are believed to be the main vector of introduction of very well-known invasive species such as the blue crab or the Chinese crab or the zebra mussel or the Asian clam, while biofouling is believed to be the main vector of introduction of several invasive algae species and also sessile fauna such as tunicates and bryozoans. But how are these species identified? Usually they are identified, or they have been mainly identified through the use of traditional approaches that use the morphology in the identification of the species. However, these approaches are highly expertise demanding. They also produce inconsistencies and inaccuracies among taxonomists because there is no master key for all species. Different taxonomists use different keys, which can lead to different results. They are also highly time consuming, and they tend to focus on specific taxa because taxonomists are not able to identify all taxa. And because of this, they are also limited in space and time and have a low throughput because we cannot analyze many samples at a time. They also can lack taxonomic resolution for very similar morphospecies, and they are also very difficult to use in the identification of small organisms such as microalgae or zooplankton, and in the identification of species in life stages beyond the adult stages such as propagules or planktonic larvae or eggs. On the other hand, DNA-based tools are morphology and life stage independent. They also have increased sensitivity because they allow the detection of low biomass species, such as the case of the recent introduction non-indigenous species, which is a priority for the early detection of these species. They also have high taxonomic resolution because they enable differentiation of very similar morphospecies, and they also allow the concomitant monitoring of a wider taxonomic range of species. They need less training, at least less than the one that is needed to produce a good taxonomist. And because of this, they can be faster, they can have high throughput because we can analyze more samples at a time, and they are also easier to automate. In particular, DNA metabarcoding is one of the most promising tools for the early detection of non-indigenous species in coastal ecosystems. And in this technique, DNA is extracted from a mixture of organisms or from environmental DNA, such as water, for where the organisms can release their DNA. A gene fragment is amplified using universal primers. Here I have represented the 18S and the CO1, which are the most used for the detection of animal species. Then the sequences of these species are produced by next generation sequencing or high throughput sequencing, which can generate thousands to millions of sequences per reaction. And then the identity of these sequences is obtained by comparing to those sequences that exist in reference databases. But although this technique seems so promising, are we ready to monitor non-indigenous species by using DNA metabarcoding in coastal ecosystems? In order to answer to this, we made a review in literature on publications using DNA metabarcoding for detecting and monitoring eukaryotic non-indigenous species. But what did we analyze in the current review? We made a comprehensive compilation of all the methodologies employed through the analytical chain of the DNA metabarcoding, which include the sampling strategy, such as the substrate sampled, also the biological targets, the DNA extraction protocols used, the marker lossy and the primer pairs, the sequencing platforms, and also the bioinformatic pipelines employed. At the end, we also made a SWOT analysis by evaluating the strengths, the weaknesses, the opportunities, and the threats of the DNA metabarcoding approach in comparison to the morphology-based approach. So we found 42 publications in literature using DNA metabarcoding for detecting and monitoring eukaryotic non-indigenous species. Here is our paper. It was recently published. In order, if you want to know more about this work, you can look at it. These publications were published between 2013 and 2020, and most of these studies have been conducted in Europe, in particular in Spain, also in Australia and New Zealand, North America, while a few studies have been conducted in Asia. And no studies at all were found in Africa or South America. Concerning the substrates and biological targets, it depended a lot on the objective of the study. For instance, seawater and ballast water were the most assessed substrates, and on these substrates, the most assessed biological targets were zooplankton, here represented in yellow, or eDNA, the environmental DNA, represented in light blue. In phytoplankton, which can include diatoms and microalgae that can form toxic wombs, were namely monitored on ballast waters or on sediments, while benthic invertebrates in sediment, here in orange, and falling eukaryotes such as bryozoans or tunicates, namely the sessile fauna, were highly monitored in settlement plates, which are plates, artificial plates that are deployed on sites for defined periods of time. However, since different species can have different non-indigenous species can have different habitat preferences, and they have different life cycles. Different sampling, different substrates should be done in order to maximize non-indigenous detection. However, only 20%, around 20% of the studies sampled more than one substrate. And most of these substrates and biological targets were sampled in ports and marinas, and also in ship ballast waters, because these are priority hubs to detect early non-indigenous species in coastal ecosystems, because it's where ships arrive and can release non-indigenous organisms. Concerning the DNA extraction protocols, most studies have used DNA extraction kits. These have the advantage of having shortened isolation steps, also handling of less toxic chemicals such as phenol, and the major disadvantage is that they can be a bit cost, but they also yield high quality DNA. And most kits are very similar. The major difference is, and that can make a difference on DNA extraction, on the efficiency of DNA extraction, is the lysis method. For instance, for animals, heat, chemical, or enzymatic lysis can be enough. But for instance, for phytoplankton that include microalgae and they have harder cell walls, maybe a mechanical lysis it's preferred in order that the DNA extraction is more efficient. Concerning the marker lossy and primer pairs, the 18S and the CO1 have been the most widely used. The 18S is the small subunit of the ribosomal RNA gene, and the CO1 is the marker that's used for the DNA barcoding of animals, and it is the gene that codifies for the subunit 1 of the enzyme cytochrome oxidase mitochondrial. And both markers have advantages and disadvantages. For instance, the 18S usually has high amplification rates because the primer binding sites are more conserved, but this primer can lack taxonomic resolution for many similar species. On the other hand, the CO1 marker sometimes has low amplification success, but we can achieve with this marker many identifications at species level. However, different primers have been used, with difficult comparisons among studies. Concerning the sequencing platform, the Illumina MySeq has been the most widely used. And in fact, this platform provides a good compromise between the sequencing depth and also the low sequencing error rates. And there are already well-established bioinformatic procedures by this platform. Usually, the most used sequencing depth is around 6,000 reads per amplicon. This means that each amplicon is read 6,000 times in the sequencer, and increasing the sequencing depth can be useful for detecting rare species, such as the case of recently introduced non-indigenous species, but it's a bit, it can become more expensive. Concerning the bioinformatic pipelines, so the pipelines that are used for processing the sequence data, usually the sequence data after sequencing is processed in 4 main stages. The first two for removing the primer sequences, for removing the sequencing errors. The third one, usually in order to reduce a bit our data set, the sequences are grouped in operational taxonomic unities, which are OTUs, based on sequence similarity, usually between 97 and 99%. And then the last step is the taxonomic assignment of representative OTUs, or sequence variants, which are becoming widely used, where the sequences are not grouped in OTUs, and it's the taxonomic assignment against reference databases. As you can see, several different algorithms have been used on each of these steps, and I put here only the most used, not even all that are used. And fortunately, nowadays, there are already platforms such as the Embrave and the Silva NGS that where you can process the sequence data, and they are very user-friendly. At the end, what we want to obtain, it's a table, an OTU table, where you have the OTUs, you have the identifications at species level, the percentage of identity, the number of reads per sample, and this table can be used for downstream analysis, such in every ecological study. Concerning the taxonomic assignment, this is usually conducted by blasting the OTUs or the exact sequence variants against genetic reference databases. You have the gene bank, which is one of the most used and is the one that has a high number of sequences, but it's also the one that it's more prone to errors because it's not curated. On the other hand, you have the barcode of life data systems, the BOLD, which is used namely for depositing data from barcoding studies. And it is a very good repository for CO1 sequences, and it is better curated than GenBank because the sequences, for instance, for being considered barcode compliant, they have to pass several filters. And you also have PR2 database or the Silva database that are widely used for blasting 18S sequences. However, and this is one of the major issues that can affect the diversity recovered through DNA metabarcoding, is that reference libraries can be incomplete, and this can really affect the assessed diversity. And some authors, what to do, some researchers, what to do is to construct custom local reference databases where during their experiments, they also collect specimens and generate DNA barcodes for those specimens. And these authors here, this study, Abad et al. From 2016, they were able, by doing a custom local reference database, of increasing the taxonomic assignment from 20 to 50%. This study here, also Couton et al., they were able to increase the success of non-indigenous species detection by assembling and by filling a custom local reference database that were missed when they conducted the blast against the gene bank. And in fact, reference libraries are still highly incomplete. For instance, 30 to 50% of the non-indigenous species occurring in Europe still miss DNA barcodes. And also the completeness of these reference libraries varied among taxonomic groups and markers. For instance, for animals, most taxonomic groups are better represented by CO1 sequences, represented in orange, but for instance, some groups such as the ctenophora, the platyhelminth, or the porifera, they are better represented by 18S sequence, represented in yellow. Also, for the chromista, most groups are better represented by 18S sequence, but for instance, Theocrophyta, which include diatoms and brown algae, are better represented by RBCL sequences, represented here in green. So, while reference libraries are far from completion, maybe using different markers can be a better approach, but only 31 of the publications used at least 2 markers. And now to complete these reference libraries by doing DNA barcoding. During the DNA barcoding, the DNA is extracted from a tissue sample from a specimen. The DNA barcodes are produced by PCR and by using primers targeting one of the regions formerly used for each biological domain, for instance, CO1 for animals, also for macroalgae and for some chromista groups. The genes codifying for maturase K and rubisco, which is the gene that codifies for the large subunit of this enzyme, for plants, and also for some groups of chromista, namely diatoms, and the small subunit ribosomal DNA, the 18S, for chromista, in particular for protists. But since animals are also widely targeted DNA metabarcoding studies by using this marker, sequences belonging to this marker for animals should be also generated. And then the DNA barcodes are deposited in genetic databases with some other information, such as data collection of the specimen, also photos. And the most widely used genetic database is the Barcode of Life Data Systems, the BOLD. And for finalizing this part of the presentation, I will make a SWOT analysis where I will present the strengths, the weaknesses, the opportunities, and the threats of the DNA metabarcoding to be used in bioinvasion ecology in comparison with the morphology-based approach. And I will start with the worst, with the weaknesses and the threats. And so, as you were able to see during the presentation, in fact, there is a lack of standard protocols. The protocols vary a lot through the analytical chain of the DNA metabarcoding approach, which made the comparison among studies hard to accomplish. Also, the lack of universal primers, and with this I mean a primer pair that amplifies the DNA of all species in a mixture, or the incompleteness of reference libraries, also the presence of errors in these reference libraries, such as misidentifications, can lead to false negatives, which mean that a species may be present but not detected. Also, the impossibility to quantify abundance, difficult to evaluate the population status, and this is very important to evaluate the population status of non-indigenous species, because it's very important to know if the species is established or not in the ecosystems. But we also found several strengths and opportunities of the technique in relation to the morphology-based approach. It is faster. It also detects more taxa. It allows the detection of non-indigenous species in all life stages. It has high sensitivity, such as in detecting low biomass species, and it can also have a higher taxonomic resolution for very similar morphospecies. This gives us the opportunity to early detect non-indigenous species, to monitor a wide taxonomic range of these species with minimum training. When I say minimum training, minimum taxonomic training. And this, in fact, can be more responsive to regulatory and immediate management needs. They also allow high throughput since more samples can be analyzed at once. And they also allow the production of high density data, both spatial and temporal, which can significantly improve the resolution of surveillance programs. And it also seems to be a promising tool to be employed in ballast water management, in particular to assess the effect of treatment procedures. So, answering to my initial question, if we are ready to monitor non-indigenous species in coastal ecosystems through DNA metabarcoding, I think that DNA metabarcoding is ready to be used as a screening or complementary method to detect non-indigenous species, where positive detections can trigger more targeted molecular approaches such as quantitative PCR or morphological analysis more in depth. But continued research should be conducted in order to understand and reduce the nature of false positives and false negatives, because they both can trigger action or inaction when not needed. And this should be done by using, those can be more or less understood by using controls throughout the analytical chain of the DNA metabarcoding, such as negative controls, positive controls where DNA from known species is used. Also the use of mock communities, which are communities with known compositions, and also the comparison with morphology in order to and this is very important, in order to benchmark the DNA metabarcoding with the morphology-based approach. And in fact, most studies have been done, in particular negative controls, in order to flag the false positives. Nowadays, we heard a lot to talk about false positives because of the COVID-19 tests. And in fact, these controls are very important. On the other hand, less studies have been using positive controls. Also, reference sequence database should be improved and also curated to flag the presence of errors. And standardized protocols should be developed. And all together, this will facilitate the uptake of DNA metabarcoding for routine non-indigenous species surveillance in aquatic ecosystems. But what about Portugal? In Portugal, we have reported that around 180 non-indigenous species. This number has been increasing over the years, and most of these species belong, we have many red algae. We also have many mollusca species, chordata, in particular ascidians, arthropoda, in particular crustaceans. And among these species, we have 46 high impact species that are species that are already very well established, and they are invasive. We have here several invasive algae, green algae, red algae, brown algae. We have several molluscan species such as the Corbicua fulminea, which it's a very problematic species. We have Crepidula fornicata, which has been introduced and I think reported for the first time in 2016, but it's also an invasive species. We have tunicates such as Stiella plicata and Corella eumiota, which in particular Stiella plicata can interfere with bivalves production. And we have also bryozoans. We have the Chinese crab, which is a very high impact, a very problematic species that interfere with fishing activities, predate several native species. We have barnacles, we have the bull crab, and we have also the Procambarus clarki, which is the Louisiana crab, which is also a very problematic species. And 13 of these species, only 13 are shared among the three regions of Portugal, the continent, Açores, and Madeira. And 30% of these species, which are represented in this figure in red, still miss DNA barcodes in both bold and gene bank. And 10% are singletons, which means that they are represented by only one single sequence, and so more sequences should also be generated for these species in order to increase reliability of the assignments. And we have a project, the NIS-DNA project, which aims to detect early and monitor non-indigenous species in coastal ecosystems based on high throughput sequencing tools. It is funded by FCT, and it has a multidisciplinary team composed by us, by the CBMA that are leading the project, the CBWINB from University of Açores, the MARE from University of Lisbon, and we also rely on the consultancy of the University of Guelph. And what we have been doing, we have been identifying the gaps on genetic databases for the species occurring in Portugal, but also at European level, because are species that can be introduced anytime in Portugal. We are also generating DNA barcodes for non-indigenous species occurring in Portugal. We are sampling in 9 marinas in 6 geographical locations, and by using different sampling methodologies in order that we can increase the chance of detecting these species. These are the regions that we are sampling: Viana do Castelo, Porto, Aveiro, Lisboa, Madeira and Açores. We are sampling the water for environmental DNA analysis, where we are sampling zooplankton in order to detect juveniles or early stage of these species. And we are also sampling on the hard substrates, such as the pontoons, the ropes, the buoys from the marinas. And we are also optimized DNA extraction protocols, because different organisms may need different DNA extraction protocols. We are also testing primers targeting different markers, in particular CO1 and 18S. And we are also devoting a lot of time in results analysis. And I will show you only some preliminary results. By now, and by using only the CO1 marker, we were able to detect in these 3 regions, Viana do Castelo, Leixões and Aveiro, 24 non-indigenous species, namely crustaceans, mollusca, bryozoa, but the novelty is that we were able to detect 4 species that are not yet recorded in Portugal. So DNA metabarcoding is already proving its efficiency in detecting early species that were not detected before by using morphology-based approaches. And so at the end, if in fact we are successful in developing a tool, we want to devise a framework for its implementation as an early warning system for the detection of non-indigenous species in coastal ecosystems. And I want just to finish by acknowledging all the NIS-DNA team members. It is a large team, but I want to thank in particular to the CBMA members that are devoting more time in optimizing these methodologies, in particular to Felipe Costa, which is the co-PI of the project. It's also the leader of the Metabarcoding group where I work. Pedro Vieira, which is a researcher that is almost since the beginning of the project. He participated in the first samplings. Also is devoting a lot of time in optimizing bioinformatic pipelines. Ana Sofia Obrador, that she joined the project almost 1 year ago. She joined in a very hard moment because she entered the project and the university closed because of the COVID-19 outbreak. And but she embraced the challenge, and she's even starting a PhD under the scope of this project, and she is conducting almost all the lab work. And I hope that in the future she will be able also to present the results in a Science on Screen seminar. And also to Fabio Amaral, which is a master's student, also doing his work under the scope of this project, is namely analyzing the samples from Leixões. And also to Ronaldo Souza, that it was Ronaldo Souza that challenged us to use these tools to monitor invasive species in coastal ecosystems. And thank you for your attention. And I want to I finish here with a photo of the sampling, always to sampling during these hard times. And I also want to thank to FCT and to all the marinas that, in fact, are providing all the conditions for us to sample. And thank you.
Thank you very much, Sofia. It's now open. We are now open for questions for Sofia. Does anyone have?
So you're able to unmute yourselves if you want.
Yes. I just, well, to kick off the questions, I'd like to ask you, Sofia, whether all the non-indigenous species have the potential to become invasive species, or when you speak about non-indigenous, you are already meaning invasive. Of course, they are invasive if they are not they are not indigenous. But how are all? Maybe I can formulate the question in another way. Are all of them bad? I mean, couldn't they contribute in some way to the diversity? Or I'm just saying nonsense.
No, they are not all bad. Not all are invasive, in fact. Those that are invasive are, in fact, those that proliferate very rapidly and grow very fast and become invasive and interfere a lot with the native species. But not all the species are bad. In fact, there are some that are introduced by human. Well, but that introduction is controlled, of course, is maintained in aquaculture, but there are some species that are introduced for use in gastronomy, for instance.
Okay, so and when I see that the early detection of non-indigenous species is very important, of course, to prevent um, the spread. But what measures can be taken? I understand that, for instance, in ballast water, the water can be treated if before being released, if non-indigenous species are detected. But for instance, in the others that come into the ships attached to the ship, how can you control that? How can it be controlled?
Yeah, well, the best way it's to avoid its introduction, of course. With the ballast waters, there are several measures nowadays that entered in force in 2017, I think. In Portugal, they became in vigor in 2018. And these waters, what the measures are that in fact, ballast waters are released far away from the coast, at least 200 nautical miles, I think. Also, there is now a time, I think that 5 years, for the ships to have a system to sterilize the waters, such as UVs or other system, since they do not put at risk biodiversity, of course. And with the ships, it's more difficult, but and I don't know if there is a measure, but at least with the recreational boats, they should be cleaning, because the recreational boats are a way also of spreading the species through the ecosystems. And with the recreational boats, I know that they have to make constant cleanings of the ships and not in the water. Outside the water, they have to do those cleanings. And the funny thing is that when we were sampling in marinas, in fact, they were saying ships were those that should be cleaned, because in fact they bring the species. But I think there is not a measure, but I know that with the recreational ships, they obligate to clean the ships.
Okay, does anyone have questions? No, I don't see.
If someone has questions, can always contact me, of course.
So you are available to
Yeah, of course.
And to clarify some points that. But well, I think you did a great job. So it was all clear, and you have really all this compilation of works. It's very important, really. You are doing a great job. Congratulations.
Thank you.
Thank you, and thank you everybody for participating. And I'll see you all in 2 weeks for our next seminar.
Okay. Bye bye then. Thank you.
Thank you, Sofia. Thank you all. Bye. Take care.